Without oxygen, you can't carry out the Krebs cycle. Pyruvate builds up within the cell, and as it builds up, it gets converted to lactic acid, which then enters the bloodstream. You develop what's called a metabolic acidosis. That's characteristic of all forms of shock. The tissues do not receive enough oxygen. They can't carry out the Krebs cycle. Pyruvate builds up and gets converted to lactic acid, which is released into the bloodstream. And so it's going to alter the pH of your blood. That's a characteristic of shock. Or what happens in muscle. Why is it that muscles fatigue? Well, because at a certain point, the exercising muscle requires more oxygen. The demand for oxygen is much greater than its supply. And so when you don't have an adequate supply, pyruvate builds up, you form lactic acid, and that, as it accumulates within the muscle cell, contributes to fatigue. Yes?
What do you call it? With a lactic acid, like it's the metabolic
Acidosis. Acidosis. You know, lactic acid is like any other acid. If it's released into the bloodstream, it releases hydrogen. And as the hydrogen ion concentration in the blood increases, the pH drops. It's the scarecrow effect. Okay? As hydrogen ion goes up, pH goes down. All right, and this is the sum total. If we were to summarize all of those steps involved in glycolysis and the Krebs cycle, okay, and summarize that reaction, it would be 1 glucose plus 6 oxygens. Okay, remember we need oxygen for the Krebs cycle. It's going to yield 36 ATPs, 6 waters, and 6 CO2 molecules. Now, I said that some of that ATP that's synthesized during glycolysis is used to transport pyruvate. Some cells, like the heart and liver cells, show electrons more efficiently. They can create more ATP than the 36. Like the heart and the liver can produce up to 38 molecules of ATP. Okay. Let's talk about other nutrients. Lipids. Okay. We always say that lipids represent a heterogeneous group. That means it's not all one type of molecule. There are several different molecules that fit under the category of lipids. Okay? And the common denominator is that all of these different types of molecules are soluble. They dissolve within nonpolar solvents. Classic example is benzene. Whereas they would not dissolve in any type of solvent which is polar, like water. Now water has a positive end molecule and a negative end. Well, that makes it polar. And in that case, then the lipids aren't going to dissolve in that because they're non-polar. Like dissolves in like. Okay? But this category includes fatty acids. Okay? If we attach fatty acids to glycerol, we form glycerides, like monoglycerides, diglycerides, and triglycerides. It includes phospholipids, which are used for cell membranes, and cholesterol, which are both a part of cell membranes, but they also are used to generate steroid molecules, steroid hormones. Okay. A fatty acid is simply a long hydrocarbon chain, at the end of which is a carboxyl group. Carbon attached to an oxygen on one side and a hydroxyl group on the other. Remember from our octet rule that all carbons are going to have 4 bonds. Why? Because they have 4 electrons in their outermost shell, and they need 4 more in order to fill up that shell to complete the octet rule. Okay. So they're sharing 4 pairs of electrons. Notice that some of these fatty acids that I've demonstrated here are, in fact, all 3 of these are unsaturated. Unsaturated just reflects the fact that the carbons aren't maximally saturated with hydrogen. See, if you form a double bond here, that's one less hydrogen that could be connected to that carbon. Okay? So fatty acids differ one from another based on the length of the chain and the number and position of the double bonds. If you have only one double bond, we'd say that's a monounsaturated fatty acid. If there are multiple double bonds, we'd say it's polyunsaturated. Okay? So this is what fatty acids look like. In biochemistry, again, you would learn all the names of your common fatty acids. But this is biology. We use fatty acids to produce glycerides. Okay? Glycerides are simply lipids in which we attach fatty acids to a glycerol backbone. Glycerol is a 3-carbon molecule, and you have a hydroxyl group attached to each carbon. Well, if we add a fatty acid at the site of these hydroxyl groups, then we form glycerides. If we add one fatty acid to glycerol, we form a monoglyceride. If we add 2, it would be a diglyceride, and of course 3 would be a triglyceride. So in this particular formula here, this particular molecular structure, the R group here represents the hydrocarbon chain associated with the fatty acid. This is its hydroxyl group. And you see it's attached here to the glycerol. Now, I said earlier that there are some tissues that prefer fat. When I say fat, I mean triglycerides, okay, over glucose. Where fat is the preferred fuel source. They can use glucose, but they prefer fat. Those tissues would be the liver, adipose tissue, and that makes sense, and resting skeletal muscle. All the other tissues prefer glucose. Now, those tissues that prefer glucose, though, most of them can use fat if glucose isn't available. The exception being the brain and red blood cells. Okay? Now, we talked about forming monoglycerides, diglycerides, and triglycerides. In the body, most of the glycerides that you're going to find are triglycerides. When we ingest fat in our diet, those are triglycerides. Monoglycerides and diglycerides don't appear in any appreciable amounts in nature. Now, in our GI tract, they'll be formed briefly as we're breaking down triglycerides. As we're converting triglycerides to glycerol and fatty acids, there's going to be a brief stage where you have a monoglyceride or a diglyceride. But once absorption occurs into the bloodstream and our body reassembles, then you don't find much in the way of monoglycerides or diglycerides. It's all triglycerides. That's how we store energy. Okay. Cholesterol is another type of lipid, but it looks entirely different from triglycerides. It's got a 4-ring structure. Big old hydrocarbons with 4 rings like this. A little branch, a little side chain. You know from AMP1 that it's used to stabilize cell membranes. It adds rigidity to the membrane. And it's also used as a precursor to other important molecules in the body, like bile salts or steroid hormones. And vitamin D. Vitamin D is actually a steroid hormone. Okay. We said that there are certain tissues, adipose tissue, the liver, the resting skeletal muscle, that prefer triglycerides as their fuel source. But when dietary carbohydrates are limited, then other tissues will begin to break down fat as well for energy. In fact, that's the basis of the Atkins diet, or sugar busters. If you restrict your carbohydrate intake enough, you restrict the intake of starch, which is your main carbohydrate, then what does your body start to do? Starts to burn fat. So you're forcing it to oxidize fat. Okay. Now, what happens when we break down fat? When you break down fat, you form glycerol and fatty acids. Glycerol can be converted to glucose. In fact, when we convert glycerol to glucose, there's a name for that process. It's called gluconeogenesis. When you convert non-glucose molecules into glucose. So in that way, the glycerol can serve as fuel. But the problem is that the glycerol only represents 5% of the fat molecule. So it's not an efficient source of glucose. Okay. When we break down triglycerides, most of which what we get is fatty acids. So what the body does to these fatty acids, these long hydrocarbon chains, is to break them down into 2 carbon fragments. Okay, so we'll have a long, big old long fatty acid. And this process, by the way, is called beta hydroxylation. Or beta oxidation, I'm sorry, beta oxidation. So it's going to break down these big old fatty acid chains into 2 carbon fragments. And it's going to take each of these 2 carbon fragments and attach it to coenzyme A. So that you form then acetyl coenzyme A from these 2 carbon fragments. Okay? And then that acetyl coenzyme A, remember, can enter into the Krebs cycle. Because that's the starting point. So that's the way you use fatty acids or fat as an alternative source of fuel. You take glycerol, convert to glucose, take the fatty acids, chop them down, and convert them to acetyl coenzyme A. And now we're generating energy from those fatty acids. All right, now I put a picture in here of that just to remind you what adipose tissue looks like. This is like a blast from the past. You know, when you do something like this, everybody feels good. Everybody feels warm and fuzzy.
Yeah.
Oh yeah, I recognize that. Yeah. It's like seeing an old friend. It's like seeing an old friend. All right. Now, you know, for the most part, proteins are not broken down for fuel. Most of the proteins that we take in our diet are broken down into amino acids, which are then synthesized into either structural proteins, might be like muscle proteins, that contribute to the muscle tissue, okay, or functional proteins like enzymes or some hormones. Okay. The fact is that proteins generally are not used as a source of fuel. The only time we use proteins as a source of fuel is if times are bad. Okay? Imagine yourself, for instance, on Naked and Afraid. Okay? You're naked and you're afraid. And you've withstood the first 15, 16, 17 days. And you're out there on day 19 or 20 or 21, and you haven't done a very good job of tracking down food. Okay? And as a result, well, you're naked and afraid. But your body has begun to catabolize itself. Okay? It's begun to break down, for instance, fat. At the same time, it's beginning to break down protein as a source of fuel. Is that a good thing? No, because now you're exhibiting muscle wasting. Okay? So the body's catabolizing itself. So what happens is we take those proteins, could be muscle proteins, for instance, because that's a large source of our structural protein, and we break those proteins down into amino acids. They're basic building blocks. Remember that all amino acids have an amino terminal and a carboxyl terminal. See if you remember this. Y'all remember this?
Uh-huh.
Okay. Now, God bless you. Sometimes when we then use these amino acids as a source of fuel, many times then the first thing we do is we deaminate them. Deaminate means we break this bond here, releasing this nitrogen group. or it could be called an amino group. Okay? So the amino acids undergo deamination. Some of these amino acids can be converted to pyruvate. And if we convert them to pyruvate, then they can travel into the mitochondria and participate in the Krebs cycle. Okay? Or they can be converted back to glucose. That would be called gluconeogenesis. And saved. Or they can be converted into not only acetyl coenzyme A, but they can be converted to other intermediates within the Krebs cycle. Okay? Depending upon the nature of that specific amino acid. So then they're used as fuel. So here's what I mean. In blue here, you have about 20 different amino acids that our body utilizes. Okay, they go by different names like glycine, serine, and alanine, and cysteine. Okay, phenylalanine, leucine. I can keep going all day. But you notice that when we deaminate these amino acids, some get converted to pyruvate, some get converted to acetyl coenzyme A, some get converted to other intermediates within this Krebs cycle. Okay, depending upon their underlying molecular structure, they'll be converted to some point within the Krebs cycle, and then they're further metabolized and generate ATP. Okay? So in blue, here's your amino acids. So they get shunted into the Krebs cycle. Remember, with the fatty acids, the glycerol, the triglycerides, the glycerol component gets converted to glucose. Fatty acids can be broken down through beta oxidation into acetyl coenzyme A. So in some way, we're going to shunt these nutrients as they're broken down into the Krebs cycle. How much energy does the body need? Yeah, that's always been the question, huh? How much energy does a body need? Okay? Well, to maintain a stable, healthy body weight, energy expenditure, the amount that you burn, has to equal the amount that you take in. I think it's one of the laws or something. Now, 2/3 of